We have several panelists today. The first is Jamie McDonald. She is a genetic counselor and works with me at ARUP, but she is also a co-director of an HHT, hereditary hemorrhagic telangiectasia. I call it HHT. The Center of Excellence and an assistant professor at the University of Utah. And then we have Dr. Jordan Laser, who is medical director of Pathology and Laboratory Medicine at North Shore Long Island Jewish Health System. And then Dr. Stephen Day, who is an IVD consultant. And Jordan and Stephen are both also members of the AMP's Professional Relations Committee. And this is a topic that we've had long discussions with our PRC. And you will be able to see how come the Professional Relations Committee of AMP is such an interesting committee to be on. We basically wanted to talk about, with all of our discussions, it really always goes back to what is in the best interest of the patient. We all have one goal, and that is to provide good laboratory diagnostic testing for the patients and for our patients. But there may be different perspectives, and Jamie will represent a clinician, genetic counselor, who would be ordering such tests. Jordan will be taking the role of the innovator, and then Stephen will be taking the role of how do you do regulation and still keep innovation.
Good morning. I am a genetic counselor with 30 years experience actually recommending and ordering genetic tests, but I am also a patient with HHT, as Elaine says, as we call it, so we don't have to say hereditary telangiectasia, or hemorrhagic telangiectasia too many times. And I'm also the mother of 3 children with HHT, so I come at this indeed from a clinician's perspective, but also from a patient and mother's perspective. Hopefully it goes without saying that it is in a patient's best interest to have readily available clinical genetic testing, but also to have robust testing and to have reliable testing. HHT I've thought of as years as a poster child of sorts for the importance of molecular diagnostic testing in medical management. It's a disorder that can't reliably be diagnosed on clinical grounds, meaning physical examination and medical history, or family history for that matter, prior to preventable catastrophic complications. Yet those life-threatening complications are preventable if diagnosed early. In a nutshell, HHT is a vascular dysplasia characterized by lesions that are called either telangiectasia if they're small, tend to be called arteriovenous malformations, or AVMs if they're big. These lesions are essentially blood vessels missing the capillary connection between an artery and vein, such that arterial high-pressure blood dumps into a vein which wasn't meant to handle it with its much thinner, less elastic walls. So these lesions present risk for hemorrhage or bleeding wherever they are, but there's also some more complex complications of the disorder that are related to the shunting per se of blood through these lesions, and those complications tend to be very under-recognized by physicians caring for patients with HHT. Again, in a nutshell, there's lots of these lesions. The telangiectasias and AVMs don't occur randomly throughout the body, but they do occur in multiple organs and tissues. The most common symptom of HHT is epistaxis, or nose bleeding, which affects almost 100% of people, but it only reaches 100% penetrance, that symptom, at about 40 years of age. The average age of onset of nosebleeds is 11 years old, and very few kids or babies have nose bleeding in the first 3 years of life. On the other hand, brain AVMs are almost always congenital. They only affect about 15 to 20% of HHT patients, but if you're in that 15 to 20%, they're very significant because when they rupture and bleed, they are almost always either lethal or severely disabling. And unlike some other vascular lesions in the brain, they don't tend to present with headaches or or seizures, some symptom prior to the day they just bleed catastrophically. So I have both personal and professional experience, because I've been around for a while, dealing with what I see as the last decade or two, big obstacle to molecular diagnostics for patients, particularly with rare single gene disorders. I saw many disorders over the last couple decades that had genes identified for which clinical diagnostic testing wasn't quickly and readily available due to gene patents. I saw that rare disorders suffered most because labs just didn't have the resources, financial or otherwise, to dedicate to the licensure issues, and certainly saw this drastically affect medical management. Again, my personal experience in 1987, my daughter, first child, died of an intracranial hemorrhage related to an AVM in her brain. At that time, no genes were known for HHT. Over the next 5 years that my second and third child were born, still no genes known for HHT. No clinical genetic testing was even available at that point. So as little babies, the two of them were sedated for brain MRIs, done with contrast, and based on a 50% chance they had HHT, knowing that there was a 50% chance we were doing surveillance unnecessarily. So very soon after that stage of my family life, in the mid-90s, the genes for HHT were identified. And our lab, and also in 1995, I helped establish a Center of Excellence, multidisciplinary, because this affects many organs at the University of Utah for HHT. And along with the clinical care we provided to patients, we had a abiding interest as of about then to develop molecular diagnostics for HHT. And we spent the next decade trying to figure out the licensure issues for those genes, which really ended up being practically insurmountable. And we're finally able to start doing molecular diagnostics for HHT at University of Utah /ARUP labs almost a decade later, when the patents were finally abandoned, in large part due to our advocacy. So I have a significant fear that this decade's obstacle for molecular diagnostics for patients with rare disorders in particular, but single gene genetic disorders in general, will be prohibitive for will make it prohibitive for labs to offer testing for rare disorders, but also that it'll favor expediency and financial considerations over robustness and competency. I've watched my lab over the last decade, since we began doing testing, continually taking those negatives and identifying new genes and figuring out new ways to test the genes we already are testing. I worry that the increased regulations of lab-developed tests in particular will favor big labs without the academic research or clinical experience with a particular disorder or genes. Again, HHT as an example, it's a multi-gene disorder that in 2015, with various technologies, has a very complex molecular testing algorithm. There are gene-specific peculiarities that influence diagnostics and interpretation, and I have seen both in our own lab testing for my baby disorder, as well as other labs that are testing for their baby disorders that they do research on and focus on clinically and otherwise, that laboratory expertise regarding a particular disorder is incredibly important. In terms of the best interests of patient and clinician, I hope that we move forward with regulation that keeps and makes testing available for rare disorders. I hope we continue to have regulation that emphasizes technical and professional competency of labs, but not necessarily regulation for individual rare disorder tests. And with that, regulation that allows for research and innovation. Thank you.
My name is Jordan Laser, and I'm here to discuss innovation and how it is in the best interest of the patient. So I wanted to start out with a couple of quotes, maybe somewhat ironic quotes. Henry Ford was quoted to say, "I see no advantage in these new clocks. They run no faster than the ones made 100 years ago." And at least in popular culture, Charles Holland Duell, who was the commissioner of the United States Patent and Trademark Office in 1898, said, "Everything that can be invented has been invented." Now, fortunately for us, that didn't come into fruition, because otherwise this would probably look more like an unemployment line instead of over 900 people at the Next Generation Diagnostic Summit. So clearly, we've been able to appreciate and enjoy a tremendous amount of technological innovation in the realm of diagnostics. Was not so long ago where most laboratories were, or the workhorse of most laboratories was a qualitative PCR, gel electrophoresis, and there were more innovations and technological advances that came along. Capillary electrophoresis, real-time PCR, and now we have near absolute quantification with droplet digital PCR. And there's also been tremendous advancements in a wide range of complementary techniques as well. Microarray technology has increased in density, increased in stability, enabled us to assess a larger section of the genome with a higher resolution. And it wouldn't be the Next Generation Diagnostic Summit without talking about next generation sequencing. Now, interestingly enough, next generation sequencing is now called second generation sequencing, because innovative minds have created what we now term third and fourth generation sequencing that deliver the promise of longer read lengths, faster sequencing, and single molecule sequencing as well. And I think many of you may be familiar with this slide. In 2001, when the first human genome was sequenced, it was at the very cheap cost of $100 million. And I think what's touted very commonly in the literature these days is the $1,000 genome. And we are hovering around a $1,000 genome right now. I would like to point out that's the technical component of the genome, not necessarily the interpretive part. But clearly, I think we've all heard the term before, how this has really outpaced Moore's law, which is commonly used or referred to in the semiconductor industry. So what has this resulted in? This decreased cost has led to a dramatic increase in accessibility to these technologies. You don't have to be a mega corporation or a government in order to be able to use these technologies. And what this has resulted in extremely fertile, rich soil for knowledge generation and innovation. So I wanted to identify some way where I can quantify or visualize how innovation and knowledge generation has occurred over the past 25 years. So I did a quick PubMed search for the past 25 years, again using the terms genetic and genomic in either the title or abstract for all journals. And I think it's probably of no surprise to everyone in this room that there's been a dramatic amount of innovation and knowledge generation. I think what's important, if you could see the green line, that the best fit trend line here is exponential. Again, probably not much of a surprise to those here. We're going to come back to this graph in just a second. But that degree of innovation, we have already realized clinical applications that have been meaningful to patients. I'm just going to go through a really high-level swath across a range of clinical specialties. But in obstetrics, non-invasive prenatal screening, the concept that we can draw maternal blood to assess fetal aneuploidies is absolutely incredible technologies. And it's just another tool in our armament to be able to identify and deliver happy, healthy, beautiful babies. In the fields of genetics and pediatrics, our knowledge of how our genome can cause and result in human disease enables us to make more accurate diagnoses. And of course, we heard about the diagnostic odysseys, the unfortunate children who clearly have some kind of genetic disorder and go through a wide range of either laboratory testing, radiographical testing, non-invasive and invasive procedures that drag out over an extended period of time and cost a tremendous amount of money. Now with the advent and the use of whole exome and whole genome sequencing, at least in a portion, 25 to 50% of these patients, we're able to assess and identify a genetic cause and end that diagnostic odyssey a little bit shorter and with less cost. In the oncology space, it's really twofold. I think we've discussed quite a lot about the somatic or tumoral mutations. We're constantly learning more about the tumor mutations and how they predict either response or resistance to a wide range of new drugs to really help us achieve that personalized or precision medicine we seek. But I think also in the inherited cancer space, as we learn more about our genome and how it predisposes us to a wide range of malignancies, we can identify these patients and identify these families, intervene with management, intervene with changing their surveillance programs, and prevent these patients from getting cancers in the first place. There's been advancements in infectious disease with the rapid multiplex diagnostics. Not only has this helped patients, but it's helped hospitals and healthcare organizations to give that rapid diagnosis, change disposition in a much more timely fashion, and identify patients that require cohorting in those environments. So what's over the horizon? Obviously, there's so much over the horizon, I couldn't talk about it here today. But all you have to do is come to a meeting like this, sign up for any professional societies meeting, and if you're anything like me, you almost immediately become overwhelmed by the amount of poster presentations and platform presentations that are really showing the advancements in this field. And also, if you're anything like me, you leave these meetings feeling a little down on yourself because how could I possibly keep up? How could I keep up and bring this and offer these to my patients? Which made me ask the question, well, what's the difference? I was able to plot innovation, but what's the difference with clinical adoption? So I actually ran the same search. However, I restricted these terms to only the 13 most prominent clinical journals, which again represented a variety of clinical specialties. Obviously, the absolute numbers are different, but I think what's most telling here is that the innovation curve is one that's exponential, and the clinical adoption curve is a linear relationship. So seeing that gap in between, I asked myself, what could that possibly represent? And what I'd like to propose here is that this is really the practice of medicine. This is where we have our early adopter providers who want to treat their patients at the forefront of medicine, can come to these meetings and adopt this new technology, this new innovation, and this new information and apply it to their patient population. This is also the practice of medicine where conservative providers can sit back and wait and feel comfortable when these types of discoveries come into clinical guidelines. I think it's important to realize that in this practice of medicine, it's not only the providers. The patients have the same dogma, right? There are patients who want their management plans to be at the bleeding or cutting edge of medicine. And there are patients who want their treatment plans to be much more conservative. And again, I think it's in this gap or in this delta where those patients can find the right physician to provide the right treatment plan that they are looking for. And anytime we have patient engagement and patient satisfaction, clearly that's in the best interest of the patient. So what are some of the take-home messages? I think it's obvious. Innovation is a necessity. Innovation to date has had some serious clinical impact. Clinical adoption is slower than innovation, and I think that's appropriate. And again, I think that delta or that gap is where medicine is practiced. And as with everything in life, it comes down to a proper balance. And I really feel very privileged to be here today, to be part of the conversation in balancing the interest of the patient with innovation and regulation. So I thank you for your time, and I really look forward to our Q&A session.
So I'm the last speaker, and I get to talk on the most fun topic of the morning, and that's regulation. All of us out here in this meeting today are here because of advances in the practice of medicine, mostly dealing with molecular diagnostics in one way, shape, or form. And as everybody knows, the FDA over the last several years has come out and said that they do want to regulate what they call laboratory-developed tests. There are certain things to think about, though. Is FDA regulation of laboratory-developed tests appropriate versus their current method of regulation, which is unclear? Should it be a hybrid between FDA and CLIA or CMS regulation? Well, first of all, the FDA is defining the term improperly. It shouldn't be laboratory developed tests. It should be laboratory developed procedures, because these tests that are developed and performed in a single lab, and they're developed by a practicing individual who's highly trained, they're professional services that encompass and integrate the design, development, validation, verification, and quality systems used in laboratory testing and include interpretive reporting in the context of clinical care. This is something that's very, very different than a commercially manufactured kit that can be sold across state lines and be used by people with varying levels of expertise. This is the practice of medicine. Does the FDA have the authority under the Food, Drug, and Cosmetic Act to regulate the practice of medicine? It does not. It has never been given that authority. In fact, there are two different places in the U.S. codes that specifically state that licensed practitioners are exempted if they prepare or manufacture drug and devices solely for use in the course of their professional practice. Laboratory developed procedures, which is the appropriate term that should be used, fit that definition perfectly. Also in the U.S. code, licensed practitioners who prescribe or administer drugs or devices and who manufacture, prepare, propagate, compound, or process drugs or devices solely for use in the course of their professional practice are also exempted from this requirement to register with the Secretary of HHS. So does the FDA have the authority to regulate laboratory developed procedures? They think they do. U.S. regulations say otherwise. So is there really a problem with laboratory developed procedures? Anytime somebody proposes change in regulations, 4 different criteria need to be met. One is you need to clearly define the problem to be solved through the regulation. Has the FDA done that? They haven't even been able to show that there is a problem. They've been asked multiple times to provide examples where patient safety or clinical management has been adversely affected by the results of laboratory developed procedures. To date, they really haven't done that at all. Secondly, they have to be able to quantitatively assess the problem. The FDA can't even do that. Right now, the vast majority of patient care and patient management that's done with the results of molecular diagnostic tests are done with the results of laboratory developed tests, not FDA cleared or approved manufactured devices. They also need to be able to show that the proposed regulatory action would effectively address the problem, and they haven't even done that. And in fact, their proposed framework of regulation is actually creating more problems than it's solving, especially economic problems and the ability of smaller laboratories and hospitals and practitioners to provide high-quality health care to their patients. And then the final one is they need to demonstrate that the regulatory burden is appropriate for the societal cost imposed by the problem. They haven't done this because, first of all, they have no understanding of how clinical laboratory medicine is practiced. They don't understand that these small laboratories and practitioners cannot burden the financial cost of actually doing PMA and 510K submissions to the FDA. And they also don't take a look at what the impact is going to be on patient care and patient management. So, are there other ways to go about regulating laboratory developed procedures if there needs to be a new way at all? There are a couple of different approaches. There have been IVD manufacturing trade groups that have come out patting the FDA on the back for their attempt to regulate laboratory developed procedures. There have been other professional societies in the laboratory industry that have come out and said that the FDA does try to go forward with this, they will be taken to court. And my guess is they probably would stand a good chance of winning against the FDA. And then there are other groups that have tried to work out compromise proposals on how to regulate laboratory developed procedures. And they fall into two extremes. They fall into those that result in a hybrid between FDA and CLIA regulation, and those that are mainly CLIA regulation with just a little bit of FDA. And I'll give two examples of both of these. The first was actually taken to Capitol Hill as a proposal a couple of months ago, and that's the Diagnostic Working Groups. This is a coalition of some of the largest IVD manufacturers and some of the largest reference labs in the United States. Their proposal is predicated on several things. First, laboratory-developed procedures are fundamentally different from medical devices. And the medical device regulations are what is currently used by the FDA to regulate in vitro diagnostics, even though in vitro diagnostics don't necessarily fit in the mold of medical devices. And there I'm talking about implantable devices and things like that. Laboratory-developed procedures don't meet the statutory definition of a medical device under the U.S. Code. The FDA has not followed the legally required process to regulate laboratory-developed procedures. They're doing it by guidance documents. Guidance documents are not legally binding documents. The way they should do it under the regulatory statutes is they should do notification and comment rulemaking. This is where they send out a notification, people can comment about it. They have to publish what all those comments are and how they're being addressed by the FDA. And you can actually go through multiple rounds of this notification and comment before the final rule is issued. Well, FDA doesn't understand the laboratory testing environment. That's well known. The laboratory environment and utilization of laboratory-developed procedures is very different than the manufacturing environment of commercially manufactured kits. Laboratory-developed procedures already undergo regulatory review through CLIA and through states and by third-party accreditation. The FDA lacks the resources necessary to regulate LDPs. They even lack the resources in their efforts to currently regulate tobacco and tobacco products. So where are they going to come up with the extra people to do laboratory-developed tests? Because there are over 11,000 different laboratory-developed tests performed daily here in the United States. And then the FDA regulation of laboratory-developed tests as medical devices would be harmful to the public by interfering with the practice of medicine and eliminating or delaying access to innovative tests and technology needed for patient care. So how do they want to go about doing this? Well, first of all, they want it regulated. The Diagnostic Test Working Group wants to do this based on risk classification. And this is their risk classification: high, moderate, and low risk. And in some respects, it mirrors the FDA's definition of what's considered high, moderate, and low risk. High risk is when the result from that test is the sole determinant used to diagnose a serious or life-threatening condition, whether there will be a significant impact from a wrong result, and whether the test is not well characterized. This is how the Diagnostic Test Working Group proposes to do this. They want to create a whole new regulatory category outside of the medical device regulations called in vitro clinical tests. These will include commercially manufactured kits as well as laboratory-developed procedures. And it breaks things up into who regulates what. So the design, development, validation of a commercially manufactured kit or something that's used and shipped across state lines to one or more labs would be done by the FDA, and this would include the preparation of reagents and other materials manufactured under CGMP, as well as the testing platform itself, not just the reagents. But if you have a true laboratory-developed procedure where it's used only on the patients in a single practice or a single practitioner's geographical group of patients, then CMS would oversee the regulation of those type of tests. And that would include the preparation of reagents other than materials used in one CLIA facility, the lab SOPs, the pre-analytical process, how these IVCTs perform, and as well as how the results are reported out. The problem with this approach is that the quality systems management required for CGMP by FDA is very different than the quality system management program that CLIA imposes upon laboratories. How are you going to resolve this too? Well, the FDA has a working group that's supposedly looking at that right now. And then there's medical application, and this is what would be regulated by the states, and that's the interpretation and consultation with the physician and patients. So this actually doesn't simplify things. It actually makes things much more difficult. It actually reduces the regulatory requirements for IVD manufacturers, but increases the liability for laboratories. So instead of just having medical malpractice insurance, they have to have general liability product insurance as well, just like an IVD manufacturer. This is going to be cost prohibitive. It's going to make the smaller labs actually have to give up on laboratory developed procedures, and that's going to result in all those tests going to the large reference labs that were actually part of the group that drafted this proposal. And their tests are not any better in many cases than the locally performed laboratory developed procedures. So this is going to adversely affect the ability to provide high quality health care over the long run. Another way of doing the regulation is the proposal that was actually published last week by the Association for Molecular Pathology. And their proposal is to update and modernize CLIA regulations and have virtually all the regulation of laboratory developed procedures done under CLIA, with CMS being in charge of that. It's designed to update the existing CLIA regulations using a tier risk-based system. The proposal is very, very detailed in how they want to go about doing this. It assures both analytical and clinical validity. The clinical validity is the main part that's missing from laboratory procedures today, because CLIA only requires analytical validation, not necessarily clinical validation. It provides a distinct role for the FDA, and these will be for multi-analyte assays using algorithmic analyses. Those will be considered high risk and require an FDA submission. Whether that's a 510K submission or a PMA submission is going to be determined by the laboratory that's actually developed the test and their conversations with FDA. It provides enhanced standardization of the responsibilities of third-party accreditors. It doesn't change states' exempt status under CLIA, prohibits duplication of CLIA activities for the purpose of assuring whether a laboratory developed procedure is reasonable and necessary. And this is an important point. If CMS is if CMS is tasked with reviewing all these LDPs and determining their reasonability and necessity, which is what they do right now under CMS administrative contractors, those contractors should not be allowed to determine whether these tests are analytically and clinically valid. There's provisions for shared LDP protocols, and the timeline is 2 years to finalize the rule, another 2 years to put it into effect. And I'm running over, so I'm going to go very, very fast. These are the risk classifications that AMP has come up with. All of this is online at amp.org. There are publication requirements. The labs will be required to use a certain form developed by CMS to actually publish their LDP's description, its analytical and clinical validity data, the methodology, the date the test was put into service, and contact information to the lab. It does ensure quality because labs will have to adapt to standard format. The test will have to be submitted to CMS for review within CMS or by an accredited third-party reviewer before it's introduced into clinical service. If it's a high-risk test, the review has to be back within 180 days. If it's a moderate-risk test, it has to be back within 30 days, and low-risk tests are exempt from submission. But they are not exempt from publication. This gives you an idea of some of the high-level oversights of the LDP regulation from AMP. And then clinical validity, instead of requiring clinical trial data the way the FDA does, evidence can be used instead. Peer-reviewed literature, clinical practice guidelines, subject matter expert opinions, bench studies, case studies, clinical data, consensus standards, all of this can be used to support the clinical validity of a laboratory-developed procedure. So this proposal from AMP is the one that I like the most because it actually addresses everything. The FDA proposal doesn't address everything. The Diagnostic Test Working Group proposal doesn't address everything. This one actually does come very, very close to addressing everything. Both the development, the submission, review, the availability of public databases, decisions on whether a laboratory can offer a test and how they can appeal that decision from CMS. Post-introduction monitoring, including adverse event reporting. Also proficiency testing. It puts more burden on the providers of proficiency testing to have more transparency, because right now, if a lab misses a proficiency sample, or if that proficiency sample doesn't obtain 80% consensus, it's just reported out as non-graded. Nobody knows why it wasn't graded. So in this proposal, providers of proficiency testing will be required to notify CMS, not FDA, CMS, any complaints against it and how those complaints were resolved. And then finally, modification of laboratory developed tests and modification of FDA cleared or approved tests are also addressed in there. So this has been a very short period of time for 3 speakers. We've presented quite a bit of information, but everything that we've presented is designed to be in the best interest of the patient. Not necessarily in the best interest of the lab, not necessarily in the best interest of the manufacturer, but what's in the best interest of the patient. And so I think it's time for us to take some questions.